Good morning children. Topic is insertional inactivation. PUC vector. What is PUC vector? It is an artificial plasmid, an artificial vector which is used to transfer the gene of interest into a bacterial cell. Full form of PUC is P stands for plasmid, University of California. This vector has ampicillin resistant gene. This vector also has gene coding for beta galactosidase. Now this is the place where the gene of interest will enter. Suppose this is gene of interest. So molecular scissor will be required to cut this and to cut this. Same type of restriction enzyme will be required and it will cut and gene of interest will enter here. Once the gene of interest enters here then what happens? The gene coding for beta galactosidase, they become destroyed, they become inactive and gene of interest will enter here. Therefore, this process is known as insertional inactivation. This will become inactive. Now, when we are doing this experiment, two cases can be there. One case can be there that there is cut in this and this both, but the gene of interest does not enter. As these have sticky ends, it will unite and that form which we will get will be the non-recombinant form. Another case will be that the beta galactoside gene get destroys and gene of interest enters and that form will be known as recombinant plasmid. Now, this has to be transferred into the bacterial cell. So, three cases can be there. In one case, none of the plasmid enters, nor this one, nor this one. Another case, the non-recombinant plasmid will enter. So, that plasmid will have the ampicillin resistant gene and also the gene coding for beta galactosidase. Third case can be that the recombinant plasmid enters carrying gene of interest, but it will have ampicillin resistant gene. Now, how to identify it practically? We require only this one. So, culture will be done. How will culture be done? In a petri dish will be taken, nutrient mediums will be applied and then in that ampicillin will be added. As this is not having any plasmid, this bacteria will not survive, it will die. As this one non-recombinant type, it has ampicillin resistant gene, so it will also survive. This transformant and this transformant both will survive because both is having ampicillin resistant gene. The recombinant and the non-recombinant both will survive. Now, how to identify these two? So, for that X-gal chemical is added in the same petri dish. What is the property of X-gal? X-gal, when it comes in contact with beta galactosidase, it turns blue. So, here the bacteria culture being done, the colony will develop blue color, colony of bacteria will grow. And here colorless bacteria will grow. We will come to identify that this is the one where beta galactosidase gene has been destroyed and gene of interest has entered. We will collect this. For further experiment, it will be transferred either into a plant cell or an animal cell. Thank you, children.